Bismillahirrahmanirrahim. Start with the name of Allah, who is the most beneficent and the most merciful. Assalamu alaikum, dear students. How are you all? Hope you will be well and hearty. This is Safiya Abdo, your botany teacher from Hira Public Higher Secondary School, Sakkar. Back with another video lecture. Students, in our previous lecture, we have completed plasma membrane and its functions. Today, in this video, we are going to start fluid mosaic model of plasma membrane. Plasma membrane is not a fixed or rigid structure. It's just like an ocean, and in the ocean, there are some boats. Boats are floating in the ocean. Ocean water is not fixed. It is always fluctuating. So boats are also fluctuating and moving from one place to another. The same thing is also present in plasma membrane. It's a dynamic structure. The fluid mosaic model explains various observations regarding the structure of functional cell membrane. According to this biological model, there is a lipid bilayer in which proteins are embedded. The lipid bilayer gives fluidity and elasticity to the membrane. Small amount of carbohydrates are also found in the cell membrane. Fluid mosaic model was proposed by S.J. Singer and G.L. Nicholson in 1972. Students, question rises here that why is it called fluid mosaic model? Fluid mosaic model was a working model of plasma membrane. In this model, lipid molecules are present in a fluid state, capable of rotating and moving laterally within the membrane. Phospholipid molecules are amphipathic. That means it would have one head and two tails. Head is hydrophilic or polar, and tail is hydrophobic and non-polar. Phospholipid molecules show two types of movement: transition movement and other is flip-flop movement. Transition movement is the movement of phospholipid molecules in the same layer. In flip-flop movements, the movement of phospholipid molecules between the two layers. Phospholipids molecules are in the form of bilayer, where tails face each other and heads are away from each other. To hold things each other, there is a weak force of attraction. This weak force of attraction is called van der Waals force of attraction. The structure and arrangement of membrane protein in the fluid mosaic model is like iceberg in the sea. The protein occur as a mosaic of discontinuous particles that penetrate deeply into and even completely through the lipid sheet. The components of plasma membranes are mobile and capable of coming together to engage in various types of transient or semi-permanent interaction. According to Singer and Nicholson, these proteins are classified into two categories on the basis of their position. Some proteins are intrinsic proteins or some are extrinsic proteins. Intrinsic protein is also called integral protein. Intrinsic protein are partially or completely embedded in phospholipid bilayer. It is permanently attached to the biological membrane. It is directly incorporated within the lipid bilayer. These proteins provide a channel through which water-soluble substances such as ions can pass back and forth between the extracellular and intracellular compartment. Moving on to extrinsic protein. Extrinsic protein is also called peripheral protein. Extrinsic protein located entirely outside the lipid bilayer on either the extracellular or cytoplasmic surface exhibit a loose association with membrane surface. Extrinsic protein serves in transport of molecules and as a receptors. The proteins are arranged as mosaic within the cell membrane. For transportation through plasma membrane, there are two main processes are involved. These are passive transport. In passive transport, osmosis and diffusion takes place, and active transport. And there are other two phenomena. These are endocytosis and exocytosis. So, let's start to begin with diffusion. Diffusion is the movement of molecules from the region of its higher concentration to the region of its lower concentration. Diffusion occurs spontaneously and no extra energy is required to bring it about. A few substances freely diffuse across plasma membrane. For example, the respiratory gases like oxygen and carbon dioxide diffuse in and out of the cell. Next, osmosis. Osmosis is the movement of water molecule from the region of its higher concentration to the region of its lower concentration through semi-permeable membrane. It maintains a balance between the osmotic pressure of the intracellular fluid and that of interstitial fluid, known as osmoregulation. Moving on to active transport. Movement of molecules from lower concentration to the higher concentration by consuming energy called active transport. It is reverse process of passive transport. In passive transport, no energy is required, but active transport requires energy. Now, endocytosis. It is a process in which the cell membrane helps to take in material by unfolding in the forms of vacuoles. Endocytosis may be phagocytosis and pinocytosis. So, moving on to phagocytosis, in which solid particles are picked and ingested by the cell. For example, WBC picked up foreign bodies from the bloodstream. In this way, they destroy the harmful bacteria which enter into our body. It is also called cell-eating process. Moving on to pinocytosis. When liquid material in bulk in the form of vesicle is taken in by endocytosis, the process is called pinocytosis, which is also called cell drinking process. Exocytosis is the reverse process of endocytosis, the process of membrane fusion and the movement of materials out of the cell is called exocytosis. The fluid mosaic model is the most acceptable model of the plasma membrane. Its main function is to separate the content of the cell from outside. Students, this is all about today's lecture. I hope these all things are just clear to you. With this, we have come to the end of this video. See you soon with another video tutorial. Till that, take care and Allah Hafiz.